So the QIMR is the old history of, consistently working with these tropical viral infections which are transmitted through either to mosquitoes or other vectors, was actually very critical because it opened up the new areas of research in the institute. The program was there, like understanding these sort of unique or novel viruses. In 1964, something remarkable happened, and that event was the discovery of this Epstein Barr virus. So that actually was the very first virus that people had identified that was associated with the human cancer. So that actually was like a turning point where the interest into the viruses and cancer and immunology sort of merged all together. And Professor John Pope, who was a pioneer in that space working at QIMR, he learned about this discovery and then started looking at in detail. So literally within 3 years of discovery of Epstein Barr virus in 1964, John Pope was the first to show that if you take white blood cells from healthy people and put this virus, which is Epstein Barr virus, in this cell, it will immortalize these cells. That is, they will live forever. So that was the first very important clue of how Epstein Barr virus causes lymphomas or blood cancer. It was a very critical discovery. 97% of the population acquires this viral infection early in the childhood. So we just live with this virus and without even knowing that we are infected with this virus. It's only when either our immune system gets compromised or there is some inherent underlying, some genetic mutation happens that actually triggers could be a cancer or it could be an autoimmune disease as well, as we now know that EBV is also associated with multiple sclerosis as well. Professor Dennis Moss, who was then a PhD student of Professor John Pope, then continued his amazing work, and he was the first in the world to show what we call T cells, that they keep check on these virus-infected B cells all the time, right through your whole life. And if you remove those killer T cells through either drug or through other immunodeficiency, bang, these virus-infected B lymphocytes just go berserk. They become a cancer.
I started these experiments looking at this production of this antigen that was common to all EBV transformed cells, but all at once things started to go wrong. The cell cultures would die for completely unexplained reasons. And then by chance I presented this seminar, and as I presented the data that had in the corner the donor of the blood that provided the B cells, and the penny dropped. I could see instantly what was happening. The cells that were from the people who were immune were exclusively cultures that went on to die. Cells were activating a T cell response, as it turned out, and killing the virus-infected cells. And in those cases from the non-EBV exposed donors, they would go on to and continue to grow. And I could see immediately this was quite an important discovery. One recollection I have was I was so excited about this, I immediately went for a very fast run on Victoria Park. I was pent up with excitement, and somehow the only way I could think of getting rid of it was to run it off.
They were really making these remarkable discoveries, understanding the biology of the virus, the genetics of the virus, and then using that knowledge to really understand how the immune system works. And that was very important because that all led to development of what we now are using, what we call immunotherapy for Epstein Barr virus or any other virus. So we were able to use that knowledge to really develop and manipulate those immune cells outside the body of the patient, and then to make those cells to be strong enough to be able to kill the virus-infected cells. 5 to 6 million people each year die because of different type of cancers associated with viruses. So you need to understand the burden of those diseases, and we have to continue to work and continue to improve treatments and outcome for these patients. We have amazing facility within our institute, which is called QGen Cell Therapeutics, where we can actually grow these cells in a highly, highly sterile environment. And that can be given to the patients, not just in Brisbane, but right around the world. We have We've provided these type of cell therapies to patients in Australia, Hong Kong, US, and many other countries. Not many organizations or institutes have been able to do that, even universities in Australia. QIMR is so unique. We've been able to expand that whole program to focus on other viruses, which are equally important in a space, particularly in transplantation, in cancer setting as well. So we are treating now patients with viral complications like cytomegalovirus or polyomavirus or adenovirus, which is commonly seen in young kids who get organ or a stem cell transplant. Because they're severely immunocompromised, they're highly susceptible to get these type of infections. We're also trialing these type of cell therapies to treat patients with brain cancer, head and neck cancer, but also lymphomas as well. Another research program is to develop preventative vaccines, particularly for Epstein Barr virus and also for cytomegalovirus. And we also have a very exciting program, human papillomavirus.
Science is really about great things happening to you, boring things happening most of the time, but then when things go right, you get so excited. I used to get personally very excitable when things went right. Yes. You do realise when there is an occasional eureka moment, and they're what you really live for in science. It's not going to happen every day of the week, but when it happens, you think, I've got the right career. And when I retire, I can retire and say, yep, I think I probably achieved something in my working life.
It is some special feeling that you get when you get an email or a phone call from a clinician saying, thank you for sending your cells. Your therapy saved my patient's life. And when you read that message, all the work is done for us in a sense, because it's just amazing feeling. Being able to offer those sort of treatments to the patient come from discoveries that were made in 1960s and 70s here at QIMR. So that's something we should be proud of.